All right, welcome everybody to the 2014 Joan Marsden Lecture. It's quite an event here at McGill and for the biology department, and it's our chance to bring in the best, the brightest, and the hottest here at McGill. And so hopefully we'll get a chance to, I'll introduce the speaker in a second, but it's actually my very distinct pleasure and quite an honor to introduce Professor Ron Chase, Emeritus Professor Ron Chase, who will say a few words about Joan Marsden, the lecture series, and what it means for us here at McGill. And so it's a very special treat to have Ron come back to McGill. I had to beg and plead to have him come back out of his very joyous retreats to come back and speak to us. So let's give Ron a big hand, and thanks for coming out to tell us a few words.
Thanks, Ahab. I just want to say a few words about Joan Marsden, whom we honor today with this lecture. She was a remarkable woman who made significant, important contributions to biology at McGill. She had an association with McGill that spanned more than 60 years, beginning with her undergraduate studies here, completed in 1943, and extending to her job as director of the Bel Airs Institute in Barbados, a position she held at the time of her death in Barbados in 2001. Following her death, a fund was established to perpetuate her memory by means of these lectures. After earning a PhD at Berkeley, she returned to McGill to take up an appointment in the Department of Zoology. She proceeded to work her way up the academic ladder, lecturing to legions of pre-medical students in comparative anatomy, hence an appropriate setting here today, and conducting research on marine invertebrates. Shortly after the Bel Airs Institute opened in 1961, she and John Lewis, also of the zoology department, were the first McGill professors to exploit the research opportunities at the Institute. She became strongly attached to the people and the lifestyle of Barbados, and she returned there just about every year. Her favorite research subject was a polychaete worm, which she studied from ecological, physiological, and neurobiological perspectives. Joan was chair of zoology in 1969 when the Department of Biology was formed by the fusion of zoology, botany, and genetics. She was not only instrumental in creating the Department of Biology, but she played a major role in shaping its future through her unfailing judgment and strong character. Her accomplishments as scientist, teacher, and administrator are especially noteworthy because they were achieved during an era in which there were few women academics at McGill. As a pioneer and role model, she led the way for the women who followed her and who today enrich our department. That's part of her legacy that we celebrate today. So now Ahab will introduce the speaker.
Thanks, Ron, for that wonderful. Oh, sorry. Thanks, Ron, for that wonderful few words about Joan Marsden. Okay, so now it's my special, honor to introduce Armin Moczek, Associate Professor Armin Moczek at the University of Indiana, Bloomington. Armin is very well known for his studies on developmental plasticity, gene-by-environment interactions in quite an amazing system that he's going to talk to you today about the horns and beetles. And beetles, of course, for any of you who are interested in evolutionary biology, know hold a special place in evolutionary thought for their bewildering diversity. Armin started his journey in science at the Julius Maximilians University in Würzburg, where he did his undergraduate and his master's, and he finished in 1996 under the famous myrmecologist and behavioral biologist Bert Hodobler. He then, from there, moved on to Duke, where he got his PhD in 2002. And Armin and I were PhD mates for several years at Duke, and I can attest to Armin's excellence. And I kept joking around always that Fred Nyhout, who at some point worked on sepsid flies and dung beetles, had some sort of natural affiliation to dung in some way. So you'll hear a lot about dung today and what it can tell us about evolutionary biology. And then finally went on to do his postdoc with Lisa Nagy and Diana Wheeler at the University of Arizona, where then he ended up again at the University of Indiana in Bloomington. So it was a special, really, a special experience to have Armin as a PhD mate and to see him have done so well and rise so high in the field of evolutionary biology. Thank you. Let's give him a big hand.
Hey Ed, thank you for that wonderful introduction. Thank you all for coming. It's terrific to be here. I feel greatly honored and privileged. I had a great day meeting people and learning about the really wide diversity of research that goes on here. I'm looking forward to talking to more of you later today and maybe a little bit even tomorrow morning. Now it's my time to tell you about what I do. So what I would like to do is, over the next 45 minutes, is to give you a flavor of the research that goes on in my lab. All of which, one way or another, revolves around the same question: the origin of novel complex traits in development and evolution. And much of it, in fact, not all, but much of it, and all I'm going to talk about today, revolves around the origin of horns and horn-like structures. Curiously, that question, the origin of horns and horn-like structures, seems to have captivated evolutionary biologists ever since inception of the discipline. And more than 200 years ago, in 1809, Jean-Baptiste Lamarck had this to say about the origin of horns and horn-like structures, and I quote, "In the frequent fits of anger to which the males especially are subject, the efforts of their inner feelings cause the fluids to flow more strongly towards that part of their head. In some there is hence deposited a secretion of horny matter, and in others of bony matter mixed with horny matter, which gives rise to solid protuberance. Thus we have the origin of horns and horns." Okay. So, if you take nothing away from my talk today, the one thing I would like you to take away from it is that even though I think we owe great credit to Lamarck for his many contributions to biology, with respect to the origin of horns and horn-like structures, he cut a few corners. Okay. But before I get there, I would like to step back for 2 slides and 2 minutes and explain to you why you should be interested in the origin of novel complex traits, and why anyone like me would like to or try to address that issue by studying the horns of beetles. This is what it boils down to. Evolutionary biologists, I would argue, over the last century and a half, have developed several powerful frameworks for understanding the process of evolution. In introductory biology classes, we highlight the roles of adaptation, or of historical accidents, or of developmental constraints in shaping why organisms look, behave, function, work, are put together the way they are and not some other way. And it's largely because of these frameworks that we're in a good position to understand what it takes ecologically, developmentally, genetically, for a certain amount of evolutionary change to occur over time. What do I mean by this? I mean that ecologically, we have a framework for understanding how adaptation can drive diversification, as in the diversification of leg types across insect orders. Developmentally, we have a framework for understanding how changes in the activation patterns and timing of patterning elements can generate diversity, as in the diversification of butterfly wing patterns. Genetically, we have a framework for understanding how events such as gene duplication can enable subsequent diversification, as in the diversification of eye types across the metazoans. I would also argue, however, that despite all this progress, we remain largely in the dark with two crucial questions necessary to be answered to understand to receive a full understanding of the origins of diversity. Number one, I would argue that still we know very little about the very origins of novel complex traits. What does it take, ecologically, developmentally, genetically, not to make a leg a little longer or a little broader or a little stronger, but to evolve that first leg, that first outgrowth, that first limb bud from a previously limbless ancestor? How do you get that first pigment onto a previously unpigmented wing surface? How do you evolve that first eye, that first photoreceptor, however primitive, from a blind ancestor? How do you get innovation started? Secondly, I would argue that for the vast majority of trait classes, we know very little about the interplay between ecological, developmental, and genetic mechanisms in their subsequent diversification. Okay, granted, the behavioral and ecological significance of different leg types is pretty well understood. But the developmental genetic properties or mechanisms that allow different types of legs to develop in different taxa, or even along the body axis of the same individual, are still really poorly understood. Inversely, we actually do know a lot about what it takes to make a spot bigger, smaller, or shifted across the wingscape, but the ecological and behavioral mechanisms that drive such changes in nature and natural populations are still very poorly understood. To me, the origin of novel complex traits and the interplay between ecology, development, and genetics in that process and in their subsequent diversification continue to represent two of the greatest frontiers in evolutionary biology. And my goal as a biologist is to try to work towards these frontiers. And what I would like to do today in the next 35 minutes or so is present to you a couple of approaches that we take in my lab that hopefully, cumulatively, over the years, will allow us to make such a contribution. Specifically, I want to convince you that the study of beetle horns and horned beetles offers some unusual and surprising opportunities to get at these issues, primarily for four major reasons. Number one, beetle horns are major structures. There's nothing subtle about them. They're not like bristles on the rear end of a fly. Instead, they dominate the phenotype of their bearer, not just morphologically, but also behaviorally, where they function as weapons in male-male competition. And they help define the behavioral ecology of individuals and of populations. Beetle horns are inordinately diverse, both within and between species, and we will get to this point several times over the course of today. But most importantly, beetle horns lack obvious homology to other structures in insects or non-insect arthropods. Horns are not modified legs, mouth parts, antennae. Instead, they exist alongside these structures. And you can therefore look at them as sort of an example of an evolutionary invention that beetles came up with at some point during their history, and which since has undergone one of the most dramatic trait radiations in the animal kingdom. Right. So specifically, today I want to tell you four stories, each of which will take us progressively further back in time. We'll start out talking about how horns that exist today are made, and how our understanding of their making and development informs our understanding of their evolutionary history. We'll then take a big step back in time and explore the origins of sexual and male dimorphisms, two patterns of variation that are extremely common among horned beetles. Then we'll take another big step back in time and explore the first origin, the very first origin of horns, how they got started before they even became sexually and male dimorphic. And then for the last story, I'm going to switch gears just a little bit away from the origin and diversification of horns and towards the diversification of horned beetles, where we'll explore the interplay between developmental and evolutionary, where we'll explore trade-offs during developmental processes and how they bias, and perhaps even accelerate, evolutionary trajectories and diversification in horned beetles. Okay, before I do any of this, I need to give you a brief crash course on the whens, wheres, and hows of horn development. Here's what you need to know. Beetle horns originate during late larval development, when parts of the epidermis, shown here in blue, detach from the cuticle, and then undergo rapid cell proliferation. This is shown here in cartoon version and here as an actual sagittal section for an animal. the reason this Dapi stain is so bright is because all these cells are squished up and really dense. The resulting extra tissue gets thrown into folds, as you can see here. All this happens while the animal externally still looks like a larva, albeit a larva that's sort of lethargic and doesn't go anywhere and looks actually kind of sickly. Then the animal molts into a pupa, and horns become visible externally for the first time. And then the animal molts one more time into an adult, and you have your final adult structure. Now, for the longest time, my lab and others assumed that the reason, the developmental reason, why some individuals have horns and others do not, lies in this prepupal growth phase. Clearly, this guy has a horn because he bothered to grow one up here. This one does not because he bothered not to grow one. But the more we looked, the more often we find incidences of this. A fully horned pupa molting into a completely hornless adult. A phenomenon that we subsequently termed pupal remodeling. And I want to give you just briefly 3 examples of the power and scope of pupal remodeling. Here's the first one. This is a species called Amphiphagous binotis. Shown here is a male and a female pupa, and the male and female adult that crawled out of these individual pupae. What I want you to see is that males and females as pupae are indistinguishable. And this is shown quantitatively here as well, where I'm showing you the pupal body size on this axis and the pupal horn length on the y-axis. No sexual dimorphism. But in the adult, there is an appreciable sexual dimorphism, and that's also shown quantitatively over here. And the reason for this is that both relative and absolute horn resorption is much greater in females than in males, causing females to lose most of the structure, sorry down here, whereas males retain most of it into adulthood. So here it's pupal remodeling that generates a sexual dimorphism, not differential growth. Here's a second example, same story. Males and females make identically sized thoracic horns. There's maybe a subtle sexual dimorphism with males having a slightly longer thoracic horn. But now the inverse happens. Males resorb their thoracic horn entirely before they turn adult. Females retain it, generating one of the very rare reverse sexual dimorphisms we find in this genus. Okay, last example, Amphiphagous taurus. This is a species that's famous for its paired head horns here. I want you to ignore them. I want you to focus on the fact that they also have thoracic horns. You see one back here and here. Male and female pupae are highly sexually dimorphic, but now they both resorb it, turning themselves into sexually monomorphic adults. How crazy is that? So here pupal remodeling eliminates a transient sexual dimorphism. Okay. So to summarize that first part, all you really need to know is that horns are of epidermal origin, they grow explosively during the prepupal stage, and they're subject to size modulation during the pupal stage. Intraspecific diversity in adult horn expression is generated in 2 different ways. You can grow your horns differentially during the prepupal stage, or you can differentially lose them during the adult stage. And if you look at different species, you'll find that they actually can differ quite a bit in the degree to which they use one or the other mechanisms or combinations of the 2 to generate diversity. All right. So on to our first story. How does our understanding of the developmental regulation of contemporary horns informs our understanding of how horns originated and diversified in the first place? This is the work, or it's a large ongoing effort in the lab to which a large number of people so far have contributed. Deborah Rose, a technician who's no longer part of the lab, Bethany Vasek, my first grad student, long since graduated, Teya Kijimoto, Frank Simone, Emily Snellroot, Melissa Pispini, all postdocs. 2 of them are still in the lab, the other 2 have graduated. Motivating all of their work, at least initially, was a very general overarching observation. And that is that if you look at the way horns form, you can't escape but notice that they share many similarities with the formation of traditional appendages, such as legs and mouth parts, all of which are of epidermal origin, tend to be produced rapidly in late larval development. They just can't escape that similarity. So we used it to formulate a very general hypothesis. Maybe beetle horn development is enabled by some of the same genes and pathways that instruct development of regular insect appendages. Luckily, we know a ton about the regulation of insect appendages, especially from work on Drosophila, but also on Tribolium beetles. So there was a wealth of potential candidate pathways we could investigate. And here's a non-complete list, and don't bother reading it all, but there were, we immediately have many pathways that volunteered themselves, given what they do elsewhere, that suggested immediate functions that they might be executing in the context of horn development. There are candidate genes we could investigate. We have since worked on all of them, but I'm not going to talk about all of them. But I do want to give you one example to give you a flavor of the kinds of information you can extract from that approach, which you can learn, but also what the limitations of it are. And the example involves the LEC gap genes, which are involved in patterning the proximal distal identity of developing insect appendages. If you've never heard of these genes and the LEC gap genes, all you really need to know is that there is a set of genes which are expressed in tissue regions which subsequently turn into distal, medial, and proximal identities of a future appendage. In particular, expression of, I'll do this over here, aristolis and distalis in the tip of a forming appendage destines that part to become the tip of the appendage. Dachshund expression in the medial region specifies medial leg identity in this case, and expression of homothorax and extradentical determines where this region is going to anchor the future appendage into the body wall. So it's pretty straightforward, and you can think of them as LEC gap genes because if you mess with that function, one aspect of limb identity is missing. There's a gap. Okay. It's an interesting pathway because it's highly conserved. Any appendage you look at in Drosophila, legs, mouth parts, antennae, genitalia, they all get their proximal distal axis from this pathway. In fact, any insect you look at, any arthropod people have looked at, this same pathway is active. And subsets of it are involved in patterning projections, outgrowths across phyla, such as the toothfeeds of echinoderms and the siphons of ascidians. So why not beetle horns? First step, we looked at expression. We asked, is the transcript or protein product of these genes, is it expressed during horn development? And if so, precisely where is it expressed? And we found that for the majority of genes, 4 out of 5, it's there and it's precisely where we would expect it. Aristolis and distalis is not just in the horn, it's in the future tip. Homothorax is not just in the horn, but it's in the base, so it's extradentical. Dachshund was an outlier, it's there, but instead of being confined to a medial region, it was everywhere. But okay, 4 out of 5, not bad. Big problem though, extrapolating from gene expression to what a gene might be doing during development or its products is really tentative. And we now have many, many, many examples of genes that are expressed, and as far as we can tell, they don't do anything. Or genes that are expressed and they really do something completely different than we thought they would be doing. So we decided that we really needed to develop methodologies that allow us to test the gene function. And the most straightforward way to do this in an insect system is to see if RNA interference works. And it turns out RNA interference in these organisms works like a charm. If this is something that you don't think about every day, think of RNA interference as an experimental approach that allows you to downregulate transcript abundance or the degree of expression of a gene on a sequence-specific basis. You can isolate which gene you want to downregulate. This is what you inject. The transcript abundance goes down. And you can then look for phenotypes in response to that downregulation. And if you see some, you can link gene expression to gene function. So we went ahead and developed this methodology for Ontophagus. And we're going to give you 3 examples. This is Dachshund. And all you see here are really control phenotypes. Remember, Dachshund is that gene that is supposed to give you medial appendage identity. So if you mess with it, that medial identity should be compromised. And sure enough, if you look at this antenna, for instance, you have the distal region here in the wild type, the medial region here, and the base. That medial region is shrunk. You see the same here in the leg. They also distinct eye phenotypes because Dachshund has additional functions in eye development. We did the same for homothorax, which, as we predicted, should give us ectopic wing tissue. That was a controlled phenotype telling us, yes, it's really working. Same for distalis, which we would expect should give us an absence of distal appendage elements, which we get, they're gone. So we could now go ahead and take the animals and ask, okay, in animals in which we see unambiguous evidence that transcript abundance has been downregulated, and we see unambiguous phenotypes elsewhere in the body, do we also see effects in horn formation? And the answers have been all over the place. The answer has been no for dachshund. Despite being expressed in horns, and despite unambiguous dachshund knockdown phenotypes elsewhere in the body, there's no evidence for any phenotypes in horn development. Both thoracic horns and head horns are indistinguishable from control inject animals. Homothorax, we find a huge effect in thoracic horns and no effect in head horns of the same individuals, even though homothorax is expressed in both horn types. And then distalis, we got an effect in both horn types, but again with a twist. We did this first in Ontophagus taurus, where we saw a modest but significant effect in head horn development, but not in the thoracic horns. Now remember, taurus is that species where both males and females resorb the horn. So we replicated this in a species where at least one sex retains the horn, Ontophagus binotus, and there we did get a significant effect. We've now taken this approach and applied it to all of these pathways. We've confirmed these functions during horn development. We've published most of these results. But what's really most important to me is that we replicated this approach, in most cases, not all, across multiple species, sexes, and sometimes for different body size classes and horn types. We did this because we wanted to know, is a given function conserved, or is it variable as you look at different contexts, say different sexes or different species? And almost without exception, the answer has been, it's highly variable. There's tremendous specificity of these functions with respect to horn type, sex, or species. So collectively, these results told us that evolution of horn growth was mediated in part by the redeployment, and as we'll see later, the rewiring of a wide range of ancestral developmental processes, and possibly the acquisition of novel functions. And I will give you at least one example later on of this part. By now, this is not a big surprise. It supports a common theme in evo-devo. The origin of novel traits does not require the origin of new genes or pathways. Instead, it relies on the differential recruitment of pre-existing developmental mechanisms into new contexts. But there was more than that. We also saw that different types of horns rely on different developmental mechanisms. A horn is not a horn, it's not a horn. And they therefore may have had different and independent evolutionary histories. We also saw that little to no phylogenetic distance really appears to be necessary for the evolution of modifiers that contribute to differential expression of horns within and between species. All right. So armed with this, and I should make that point. To me, this highlights an unexpected and still in evo-devo underappreciated functional diversity that can exist within and between species. So armed with this, let me tell you where we're going next. Our next steps are to move, up until a few years ago, what we did were sort of one gene at a time studies. We want to put genes together in pathways. We're now working with entire pathways, and pathways to networks. How do pathways talk to each other? And how do changes in this conversation give you different outcomes in development and evolution? And clearly, we want to move beyond the candidate gene approach that is informed solely by what others have figured out in Drosophila or Tribolium. How are we going to get there? We've developed a growing amount of genomic and transcriptomic resources. A fair amount of this is published. A lot of it is underway. So for instance, the first dung beetle genome is completely sequenced and is now being annotated. How cool is that? And we have genome-wide transcriptomes across multiple species and multiple populations. And we're now doing this even on a body region or organ-specific basis. So with this, I would like to move on to our second study. Now that we know how horns are made, what it takes to build one, let's figure out how are they made dimorphic in the context of sexual and male morph differentiation. This is a project that is done in collaboration with Justin Andrews, a former colleague of mine at Indiana University, and Teiya Kijimoto, a postdoc. And the main motivation for this project really is twofold. Number one, actually threefold. Number one, sexual dimorphisms, and a few examples are shown over here, and male dimorphisms, shown over here, are really super common. Wherever beetles have horns, they're almost invariably sexually dimorphic, and the vast majority of species of this group, they're also male dimorphic. This is especially cool because even though it's a dimorphism either way, the determinative regime is very different. Sexual dimorphism is the consequence of sex-specific development following XY sex determination. Male dimorphism is entirely the consequence of nutritional differences during larval development. These guys could be full sips, but look very different because they have different types and qualities of food available during immature development. Lastly, there's a really cool study that highlighted that through phylogeny, in 19 of 20 instances, the gain or loss of sexual dimorphism coincided with the gain-loss of male dimorphism, as if the two are somehow linked developmentally. So we got our first breakthrough, now in hindsight, in this approach through this application of genomic resources that I hinted at earlier. Because we found that one of those 41 genes, in fact one of those 26 that is uniquely upregulated in thoracic horns and head horns, but not legs of the same large males, was this one, double sex. Now, if double sex is something you've never heard of, all you need to know really is that it's a transcription factor. You can think of it as a master regulator of somatic sex differentiation. For instance, during genitalia development, it's double sex that decides whether genitalia become ovaries or whether they become testes. That's the thing. What is cool about it is a single gene, but it is alternatively spliced. It gives you male and female specific isoforms. So one gene, but at least two products, one for each sex. So we cloned it, we generated RNAi constructs, we injected them, and here's what we found. What you can see here are control-injected small males and large males of Ontophagous taurus. They look just like wild-type individuals. They're indistinguishable. Small males have these sort of rudimentary horns. Large males have these tenfold elongated, large paired horns on their head. When we knock down the male isoform of double sex, we see no effect in small males, but the nutrition-dependent induction of horns in large males is severely compromised. This holds up quantitatively very nicely. No effect in small individuals, but a huge reduction in nutrition-sensitive horn development in large males. This tells us that the male isoform of double sex promotes nutrition-responsive horn development in males. Okay. Then we went ahead and replicated this approach in females. Females are simpler in this species. Here's a small control-injected female, a large control-injected female. Except for size, they actually look identical. Large females are just proportionately enlarged versions of small females. When we knock down the female isoform of double sex, we got the induction of tiny little horns in small males and more substantial horns in large females. These results told us that the female isoform of double sex inhibits the nutrition-responsive induction of horns in females. If we knock down both, we have males and females converging onto an intermediate form. So to summarize this part, alternative double-sex transcripts promote nutrition-sensitive horn development in males, but inhibit it in females. This is correct. This suggests that double-sex function has been recruited from regulating sex differences towards regulating morph-specific differences within one sex as a function of nutritional input. This shared genetic basis between sex and morph-specific development may therefore explain the correlated gains of losses of sexual and male dimorphism throughout Ontophagous phylogeny. But the story didn't end there. This actually, now is the time when it really caused us a headache, because we went ahead and replicated it in one more species. And before I can show you the results, I need to step back and tell you a little bit about horn phylogeny in order to explain to you, in order for why we did make sense to you. Okay, so here's a phylogeny of 48 Ontophagus species generated by Doug Emlen and colleagues in 2005. Mapped onto it in blue are all the instances of head horn development. Details don't matter. All you really need to know is that this approach found that head horn evolution occurred very early, and there's a single origin down here. It immediately involved horns placed in the back of the head, posterior head horns. Most likely, they came in pairs. And the moment there were horns, very quickly thereafter, they were expressed in a sexually dimorphic manner. This is the general rule and pattern of diversity in Ontophagus, but there's this pesky exception, Ontophagus sagittarius. And this phylogeny, Sagittarius, is the sister species, not in this phylogeny, but in the, the arrow is in the wrong place. Excuse me, Sagittarius is up here. And it's the sister species to Ontophagus taurus, the species I just told you about. It stands out from the crowd because it's the only species that has evolved a reverse sexual dimorphism, and it's the only species, or at least, at most, one of two, that has evolved a novel location of head horn development here in the front. But most importantly, for these purposes, males also have lost the posterior head horns, otherwise so abundant in this genus. Okay, so we went ahead and cloned double sex out of this species, confirmed that the molecular biology and gene structure is identical, and then knocked it down. And this is what we got. Okay, so let me try to interpret this for you, the way we did it in our publication. Let's focus first on the thoracic horns, which in wild type exist only in females but not in males. When we knock down the female isoform of double sex, we get a reduction of these horns. When we knock down the male isoform, we get the induction of not a horn but a bump. So collectively, this tells us that female double sex promotes thoracic horns, male double sex inhibits it. This is the exact same function as in Ontophagus taurus, just the sexes are flipped. Okay, let's look at these anterior head horns, which in wild type exist only in males, never in females. When we knock down the male isoform of double sex, we see no effect. But when we knock down the female isoform of double sex, we get the induction of paired anterior head horns. This tells us that female double sex normally inhibits anterior head horns. Male double sex doesn't appear to have an effect. That function is absolutely identical as in Taurus, the previous species. It just involves a new location. But the elephant in the room are these guys. Posterior paired or forked horns, which we observe both in males and females once we knock down double sex, but we never ever see them in the wild type. These data tell us that male double sex inhibits normally the expression of paired posterior head horns, which we see in all the other species and would have seen in the most recent common ancestor. Whereas female double sex inhibits the transformation of a single horn up here to a, again, ancestral character state, paired or forked posterior horns. So collectively, these data tell us that the double sex RNAi phenotypes in this species reflect a mosaic, a complicated mosaic of A, reversed functions relative to Taurus, and B, novel functions with respect to, e.g., the anterior head horns. This tells us, if that is correct, this tells us that combined double sex function in this gene appears to have diversified in this species in an extraordinarily short time period since divergence from a common ancestor about 5 million years ago. Okay, so we've learned how horns are made. We now have learned a little bit about how they're made sexually dimorphic and male-male dimorphic. How did they get started in the first place? This is work done in collaboration with an undergrad, Andrew Shelby, and a graduate student, Tammy Cruikshanks, back then co-advised by Mike Wade. And this is where the story starts. So here again is a phylogeny of 48 species of Ontophagus. On the left, generated by Doug Emlen and colleagues, 2005. On the left are males, on the right are females. And shown in green are all the instances where these species have evolved thoracic horns. And this phylogeny concluded that in order to explain present-day patterns of morphological diversity, we need to invoke at least 9 independent gains of thoracic horns in males and 7 in females. This is the most parsimonious explanation to explain what we see today. All right. But at the beginning of my talk, I showed you this species, Ontophagis binotus, which in the phylogeny is coded as having a thoracic horn in males, but not in females. But if you think back, the pupae clearly have it. It's not like the females are not able to make one. Clearly they're able to make one. They just go through the added trouble of unmaking it. So they should really be coded in green over here. Okay. The same applies for another species for which I don't have pictures, Ontophagis ecate. Then I showed you this species, Ontophagis taurus. This is a species which in this phylogeny is coded as having no thoracic horn in either males or females, but clearly it's not like that species isn't able to make them. Male and female pupae have prominent thoracic horns. In fact, they are longer than the thoracic horns we see in many species that retain them. Instead, here now, both males and females resorb it entirely. Okay. We went ahead and tried to get our hold of as many species as we could in this phylogeny that are coded as having no horns in either male and female, and then finding their pupae and asking, do we see horns in the pupal stage? And in every single case that we got our hands on, we found that the answer is yes. I also found 24 additional species of Ontophagis that are not part of this phylogeny, all of which do not have thoracic horns as adults, but they all have them as pupae. Okay. So collectively, this now raises the possibility that rather than invoking 9 and 7 independent gains of thoracic horns to explain what we see today, maybe there was just a single origin of thoracic horns. And what really diversified is a given lineage's ability to retain a horn during development or to resorb it in one or both sexes. Okay. So if this is true, then one immediate question that presents itself is, why bother? Why growing a thoracic horn when all you do with it is resorb it before you turn an adult? Why go through that trouble? You can come up with a number of hypotheses. Maybe they're pleiotropic constraints. You have to make that thoracic horn because you want to make something else. Maybe these thoracic horns are leftovers of horns that used to be an adult. I really like that hypothesis, find it intriguing. Or maybe they just have a separate function, completely independent of making a horn in the, or generating a weapon in the adult stage. It is this hypothesis that I would like to spend 5 minutes on. This is where it starts. So what you see here is a sagittal section through the head and first thoracic segment of a larva that is about to mold into a pupa. Let me explain what you're looking at. So here is the head capsule, thick piece of cuticle. Here is the first thoracic segment cuticle, thin and flimsy. Shown in blue is the epidermis, and folded up here is the future thoracic horn as it's now beginning to expand underneath the larval cuticle. I want you to focus on this region, which I'm magnifying in this slide. Whenever I fixed an animal and sliced it up at this stage, I noticed that this tip is inserting itself in the space vacated between the epidermis that has already detached from the head capsule and the head capsule itself. If I would have fixed this animal 2 hours later, that tip would have been over here, and the horn would have expanded out of the plane of the board. If you watch an animal molt head on, the first thing that breaks through the head capsule is not the head, but it is, in fact, the tip of that thoracic horn, which slices or plows open the head capsule. When you watch an animal during that process and you look down on it, you see how hemolymph is being pumped into that thoracic horn as it's advancing and how it's beginning to bulge up that head capsule, which then breaks open at these preformed suture lines. So you couldn't help but think that maybe this pupal thoracic horn is something like a molting device, something that helps shed the larval head capsule. And I need to emphasize, for those of you who don't work with insects, at this stage the animal is completely helpless. It has basically resorbed all its muscle tissue. It can't really go here and take its head capsule off. Instead, it has to swallow air and blow itself up, and then it has a little bit of peristaltic ability left, and it can pump hemolymph into selected places. That's all. So we had to test this hypothesis, right? So we developed an approach that would allow us to destroy the cells in early larval development that give rise to the pupal thoracic horn later on. So we would basically knock out the cells that generate this structure. If we succeed, we should get a pupa that lacks a thoracic horn, and if the hypothesis is correct that this is a molting organ, that pupa should still have its larval head capsule on its head, right? Does that make sense? Okay, so that's exactly what happened. We get a pupa that lacks a thoracic horn, but it retains its larval head capsule. It's not like it has a larval head under there. There is a pupal head under there. It just couldn't get out. So we replicated this approach for a number of species. This is on the Phagus binotus. Here in the phylogeny, huge effect. For the Phagus gazella, a more basal member of the phylogeny, and the species with the tiniest thoracic horns in the pupa that we could find, we can still destroy them, and it still has the same effect. And then we stepped outside the genus and the sister genus Oniticellus. There are no horns of any kind in this genus, and instead there are little bumps, but you can still do the same surgery, and when you do that, you find no effect whatsoever. So collectively, this tells us that pupal or prepupal thoracic horns may indeed facilitate the shedding of the larval head capsule. If this is correct, this would explain the maintenance of horns during development in larvae, despite the absence of horns in adults. But you still need it. This dual function of prepupal horn growth may be unique to the genus Ontophagus, and if this raises the possibility that beetle horns, at least thoracic horns, may have evolved initially as an adaptation to larval pupal development and only secondarily been co-opted to become precursors of adult traits that are now expressed in a sex and more specific manner and used as weapons to beat each other up. Okay, on to my last story. This one will be swift. Here I would like to switch gears away from how horns originated and diversify and towards how horned beetles may be diversifying, with particular emphasis on trade-offs during development. This is the work of a graduate student who recently graduated, Harald Parzer. And he and I have been interested for the longest time, in genitalia development. And I'm going to give you 3 pieces of background information that explain why he did what he did. Evidence number 1. This is a study by Kazui Kawano, came out in 2002, where he looked at reproductive character displacement in 2 species of Chalcosoma that occur both in symmetry, they overlap, and in allopatry, they don't see each other. And wherever they are in symmetry, he found they look more different from each other than when they're found in allopatry, they tend to look more similar to each other. Not a big surprise. It's actually what you would predict if there's reproductive character displacement and competitive displacement. What caught our eyes were the traits he described. Species in symmetry tended to diverge in the relative sizes of horns and the relative sizes of genitalia, specifically male copulatory organs. And what especially caught our eye was the direction of divergence. The species that in symmetry had longer horns had smaller copulatory organs. The species that had shorter horns had bigger copulatory organs. Okay, N of 1 could be a simple coincidence. Study number 2 suggested that there's little more than a coincidence. This is one that I did with Fred Nijhout. For unrelated reasons, we actually destroyed the imaginal discs that are responsible for the formation of the male copulatory organ, shown in its everted form over here. When we did that, these males had extra long horns. That was unexpected, and it suggested that genitalia and horns may somehow be linked developmentally. Changes in one bring about compensatory changes in the other. Okay. Third piece of background information. This is actually part of my dissertation, which focused on 3 populations of Ontophagus taurus, which have been introduced to different environments and have since diverged dramatically in their relative investment into horns from their ancestor. So what I'm showing you here is the scaling relationship between body size and horn length. And yellow shows the average of the Mediterranean ancestor. And that yellow is the same in all 3 panels. But see how the eastern US individuals are mostly to the left of yellow, And the Western Australian individuals are mostly to the right of yellow, and Eastern Australia is sort of on top of yellow. The cool thing about these populations is twofold. Number one, they are allopatric. They never meet. There's no opportunity for reinforcement and character displacement. Number two, we have independent, strong data that suggests that this was all driven by selection acting directly on horns. We can now therefore ask the question, or use this as an opportunity to test for correlated evolution between horns and genitalia in natural populations. So Harald went ahead and measured the heck out of these animals. And that was his prediction. We should see a negative correlation between relative investment into horns and relative investment into copulatory organs. You ready? This is what they say about beetles with long horns. These are the data we found. I've never gotten this good a correlation in my life. So when we saw this, the first thing we did was just remeasure everything, because this somehow really can't be, right? No, held up. Then we thought, okay, maybe this has nothing to do with genitalia, maybe it's just all appendages are affected, there's just something general. So we measured other traits, like say the tibia, no effect in the tibia. It's specific to the interactions between horns and genitalia. Now, what I forgot to mention is these 4 allopatric populations were founded less than 50 years ago. So we are talking something on the order of 100 generations. Well, that is striking. We wanted to know if this also holds up over longer periods of time, say across species. So Harald measured, replicated this approach across a diversity of Ontophagous species, ranging in divergence from 10,000 to 38 million years ago, and finds the same striking, tight, negative correlation between relative investment into horns and genitalia. So collectively, this tells us that trade-offs between horns and genitalia are real. We don't know why they're there, but they're there. And they leave a strong signature in morphological divergence patterns within and between species. Because we know for the Australian and US populations that selection acted on horns, given that, we know that genitalia may, under these circumstances, able to diverge as a byproduct of evolutionary changes in secondary sexual traits. This is intriguing because given the importance of copulatory organ morphology for reproductive isolation, this raises the possibility that secondary sexual trait evolution, evolutionary changes in horns, could promote evolutionary changes in genitalia for free, giving you speciation as a byproduct. Yes, this is a really speculative implication, but I still like it. Okay. If correct, this would explain how Ontophagous, that genus, managed to radiate into a genus that is famous not only for the immense diversity in horn types, but also for its immense species richness. This is the most species-rich genus in the animal kingdom, with a currently described 2,400 species in a single genus. All right. So, more generally, I hope to have shown you today that beetle horns and horned beetles offer a rich and complex microcosmos for exploring the mechanics of innovation and diversification in the natural world. As always, I have to say, we need more people to work on these organisms. If you are a graduate student looking for a project, consider them. If you're a postdoc, think about them. Apply to the postdoc that just opened up in my lab. If you're a faculty member and you're just no longer so intrigued by mice or by, I don't know, the fin limb transition or, I mean, ants, really? Come on. Check them out. 2,400 species, really, can't be wrong. Okay, I ended my talk with a quote from Lamarck. I started it, I'm going to end it with a quote from Charles Darwin, who was enamored by these animals. He loved horned beetles, and he had this to say, and I quote, "From the small size of insects, we are apt to undervalue their appearance. We could imagine a male Chalcosoma, shown here, with its polished bronze coat of mail and vast complex horns, magnified to the size of a horse or even of a dog, it would be one of the most imposing animals in the world." He's right, of course, but I also like to think that he'd be pleased to see that one of his favorite animals is now emerging as a competitive model system in Evo and EcoDevo, with gene function, genomic, and many other resources emerging. So with this, I would like to thank my many collaborators who made what you saw today possible. I'd like to thank my funding agencies, a couple of special people over the years. And lastly, thanks to all of you for your attention, and I'd be happy to take any questions.